Chapter 18. In this lecture, we'll identify the components of the urinary system and describe the system's primary functions. The urinary system structures include two kidneys that produce urine that flows through the urinary tract. The urinary tract consists of two ureters run for each kidney and a urinary bladder, which is drained by the urethra. The process of elimination of urine is termed urination. The urinary system has three major functions: excretion of organic wastes, which means removal of these waste products from body fluids; elimination, or the discharge of these waste products into the external environment; and homeostatic regulation of the blood. One of the ways that the urinary system can regulate the blood volume and pressure is by adjusting the volume of water lost in urine. And the kidneys, remember, release erythropoietin and renin. By controlling the quantities of sodium, potassium, chlorine, and calcium lost in urine, the urinary system can regulate plasma ions. Nutrients like glucose and amino acids are conserved, not excreted, while organic wastes like urea and uric acid are eliminated by the urinary system. And we'll spend a large part of this lecture focusing on these homeostatic mechanisms. Let's pause for a second and see what you learned. Now let's take a closer look at the structure of the urinary system, starting with the kidneys, which are located on either side of the vertebral column between the last thoracic and the third lumbar vertebrae. The right is often a little lower than the left, so note that this is a posterior view. Kidneys are retroperitoneal, but they're supported by connective tissue. Fibrous renal capsule covers each kidney and is surrounded by adipose tissue for cushioning. Let's look at the structures of the kidney. Kidneys are bean-shaped. They have an indentation on the medial side, which is called the hilum, which creates an exit for the ureter and the renal vein, as well as the entrance for the renal artery. The fibrous capsule covers the outer surface, and it lines the renal sinus and an internal cavity or space. The renal cortex is the outer layer, shown in light pink on this figure, and it wraps around and projects into the medulla as renal columns. The renal medulla is the darker pink inner layer that contains 6 to 18 cone-shaped renal pyramids. The tip of each renal pyramid is called a renal papillae or papilla and projects into the renal pelvis. A renal lobe is an imaginary boundary that contains a pyramid, overlying cortex, and renal columns. Looking closer at a renal lobe, urine production occurs in the nephrons, in the cortex, and pyramids of the medulla. The renal corpuscle, or glomerular or Bowman's capsule, surrounds glomerular capillaries, and filtration occurs from capillary to corpuscle. The filtrate then enters the renal tubule, which consists of the proximal convoluted tubule, to the nephron loop, or the loop of Henle, followed by the distal convoluted tubule. Nephron filtrate, aka urine, empties into the collecting duct, the start of the collecting system, which drains urine into a cup-like structure called a minor calyx. 4 to 5 minor calyxes empty into a major calyx, and major calyxes combine to form the renal pelvis. Let's look closer at the filtration membrane in the renal corpuscle. The glomerular capsule has an outer layer that forms the wall of the corpuscle and an inner layer that encloses the glomerular capillaries. These layers are separated by capsular space. The epithelium covering the capillaries are made up of podocytes with specialized cells that have foot processes that sit on the basement membrane and wrap around the capillaries. These foot processes create slits or pores through which the filtrate passes. But keep in mind that plasma proteins or formed elements like red blood cells and white blood cells do not pass through these slits or pores. The function of the renal corpuscle, like I said, is to filter plasma, but the filtrate entering the proximal convoluted tubule includes valuable nutrients, ions, and water. In the proximal convoluted tubule, the first segment of the renal tubule, a majority of the reabsorption occurs. The epithelium has transport mechanisms for nutrients and ions. Molecules are moved from the tubule to the interstitial fluid, and water will follow osmotically. Fluid in the descending loop of Henle, or the nephron loop, flows towards the renal pelvis. Here the epithelium is permeable to water, but not to solutes. The tubule makes a 180-degree turn and then enters the thick ascending limb of the loop, which flows towards the renal cortex. Here the epithelium is permeable to solutes, but not to water. This reabsorption is made possible because the interstitial fluid of the renal medulla has unusually high solute concentration. The distal convoluted tubule has three vital processes: active secretion of ions, acids, drugs, and toxins, selective reabsorption of sodium, and selective reabsorption of water. The epithelial cells of the distal convoluted tubule are called juxtaglomerular cells, which articulate tightly with the smooth muscle cells of afferent arterioles. They form a region called the macula densa, and these cells are responsible for the regulation of blood volume and pressure by secretion of erythropoietin and renin. Many distal convoluted tubules empty into one collecting duct, and several collecting ducts merge into papillary duct, which empties into a minor calyx. The collecting duct functions to adjust the final filtrate composition, determine final osmotic concentration, and to determine the final volume of urine. So clearly the vasculature of the kidneys is imperative since they receive 20 to 25% of the cardiac output. Blood flow starts with the renal artery. And then enters the interlobular arteries, which then flows into the arcuate artery, which runs along the cortex medulla boundary. This then enters the cortical radiate artery and the interlobular arteries. From there, blood enters the afferent arterioles and then the glomerular capillaries, which then enters the efferent arteriole. And the peritubual capillaries can follow two possible paths. They could enter into the cortical nephrons, and this blood flows into the peritubular capillaries surrounding the nephron tubules within the cortex, or into the juxtamedullary nephron, where the peritubular capillaries flow into the vasa recta that runs parallel to the long nephron loop deep into the medulla. The blood is then collected or drained by the cortical radiate vein, which is then drained by the arcuate vein, which is drained by the interlobular vein, and finally the renal vein. And that brings us to the checkpoint, which will end this recording.